Welcome. This is a 1 hour 15-minute cell biology lecture squished down to 10 minutes that focuses on tested material. We're doing the plasma membrane. We're going to go over the overview, the components, structure, membrane skeleton, and transport mechanisms. So when we go over to the overview, it is a phospholipid bilayer. Exterior and interior are different. You would say it's asymmetrical. And the functions of the plasma membrane are to enclose the cell and to maintain differences between the outer and inner. Notice that in the phospholipid, there is some variable R polar head. There's a phosphate group, and there's two fatty acid tails. And right where there are double bonds, there is a kink. This will be important later. So now we go to the components. You have protein, which is the functional aspect of the plasma membrane. And the functions are catalysis, which is to speed up certain reactions. You have receptors, transport, and channels, which are associated with integral proteins. So let's talk about integral proteins first. Transmembrane are the ones that go from exterior to interior. That's really all you need to know. And the lipid anchored are anchored only onto one side, and that's really the extent of their connection to the plasma membrane. GPI anchored proteins are associated with extracellular. You probably won't need to know that. And the interior or cytosolic side are typically RAS or G alpha proteins. Might be a factoid to learn. Peripheral proteins are the ones that are not attached to any phospholipids. They might be attached to these proteins. They include the nuclear lamina and the membrane skeleton, which we will get into later. Lipids. There are three basic types. You have phospholipids, sphingolipids, and sterols. So phospholipids have a choline polar head group typically, and sphingolipids have sphingosine as their polar head. Glycolipids are typically associated with ceramide, and the only real polar head groups that you would need to know might be serine and inositol. Serine is apoptosis, so think of a goldfish. How do you know it's dead? Well, it flips upside down and it floats to the top, just like with serine, because it's typically on the inside of the cell. So it will flip to the outside and a macrophage will eat the cell. Inositol is a part of cell signaling, which you will know later, but not right now. Phospholipids are empipathic, which mean it's polar here and not polar here. That's basically all it means. You have your polar head group, you have your phosphate group, glycerol backbone, and you have your fatty acid tail here. Sterols, cholesterol, the end. Carbohydrates are added onto by glycosylation. They're added to proteins, typically at asparagine, serine, and threonine, and mostly on the outer leaflet of the cell. Think of blood typing. These are just sugars. So these are all the same base, and then two different sugars added onto it. So this is typically a free response question asked on most exams. We move over to structure. Fluid mosaic model. Think of that. Fluid mosaic model. Memorize it. Whatever. Two factors that affect the fluidity of this fluid mosaic are saturation and length. So saturated and long tails. Think of bacon. Solid at room temperature. That's not really helpful for the cell. So most fatty acid tails are mostly unsaturated and relatively short, but there's a balance. It's further stabilized by cholesterol. There's a polar hydroxyl group on one end of cholesterol and a fatty nonpolar tail, and it's the bubble wrap of the plasma membrane. It keeps it from shifting around so you don't break your stuff that you order from Amazon. Remodeling is due to three things to memorize. Phospholipases, which cut phospholipids, desaturases, which introduce or remove double bonds to create or remove kinks, and acyltransferases, which just move groups onto the polar heads, possibly glycosylation. When talking about asymmetry of the plasma membrane, think of lateral shifts, you know, side to side. No casual flip-flops. This does not happen without enzymes. And flexing, which is just rotations. So you could just look at this later, pause the video if you feel like it. When talking about the membrane skeleton, think of scaffolding. You know, when you're building a skyscraper and the people are on those frames outside. The framework is spectrin for a RBC, or red blood cell. It's a dimer. It's like 2 chains. And it's just a meshwork. It binds actin, tropomyosin, and ankyrin. You're probably not going to get too extensively tested on this, but just know that the shape is due to an underlying meshwork. So now we get to the hard stuff. Transport. As you know, concentration gradients go from high to low concentration. It's spontaneous. Water is semi-permeable to the plasma membrane, even though it has a fatty inside right here. Water will still go through, but aquaporins, which are protein channels, will increase the speed at which this happens, inside or outside. Ions and large polar molecules must have channels or transporters. Ion channels have 2 basic components: selectivity filters and gate regions. The selectivity filter is somewhat complicated for most people, but all you really need to know is that there are backbone carbonyl rings, and carbonyls are associated with negative charge. Notice that potassium, sodium, lithium, they have positive charges. So, and specific radii, they're very different. So if you have a different size drill bore hole or something, then not all drill bits can, you know, spiral in. So it's the same thing here. So radius and carbonyl backbone. That's really all you need to know. Perfect size for potassium ions to get through. The gate region is kind of complicated too for most people, but all you need to know is that there are 6S residues, and there's a pore domain. So 1, 2, 3, they're normal, right? And resting membrane potential inside a cell is negative 70 millivolts. And as a action potential goes through, it's positive, 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 and it hits S4. S4 has a lot of arginine residues. Arginine is positive, as you know, and positive, they repel each other, and it'll push the pore domain open like flushing a toilet, and it allows stuff to go through. The way it stops stuff, so you know, that's this rest right here. It opens, you know, and poop or whatever goes through toilet, and it gets inactivated here by the binding of some kind of molecule here, and these inactivation peptides will block the hole until it gets back to the resting state. That was the hardest part. Yay! Types of ion channels are ligand gated, associated with allosteric. It means binding of some specific thing to another part, like a switch. Mechanogated is stretching or compression, so it's like blocking the hole physically. And voltage gated is what we just explained. So there's a charge difference and shaker channels. These are sometimes passive transport and sometimes active, but for testing purposes, think of it as just passive, because it's just a channel. Facilitated diffusion, glucose transport, it's just co-transport. Active transport, characteristics, they go against the gradient and they require energy. If it doesn't have those 2 components, forget about it. It's just passive. Light, ATP, gradients, those are the energy sources. The types, these are the major types that you're going to run across. The potassium, sodium pump, 3 in A out, 2K in, and negative inner charge. Calcium ATPases are associated with the inside of the cell in the endoplasmic reticulum, usually muscle cells, and the hydrogen potassium ATPase for stomach acid. And secondary active transport is associated with symporters or antiporters. Symport means they're traveling together, and antiporter means they're swapping. And this is the overview. Alright, so that's really it. That's all you have to know for the first lecture. Don't forget to like and subscribe, or leave comments below about things I can improve on or things that you need help with. Alright, thanks for watching.